First, thank you and welcome to everyone. Thank you for joining today's seminar on enhanced natural attenuation, pathways to sustainable remediation. The seminar today is co-hosted by the Center for Remediation of Complex Sites, or REMPLEX, as well as the Sustainable Remediation Forum, or SURF. For those of you who don't know, my name is David Peeler. I'm the director of REMPLEX. And for those of you who may be new to REMPLEX, REMPLEX was established at P&L about 6 years ago, which was really created to be a forum to foster knowledge transfer, collaborative research, professional development in the realm of environmental remediation complex sites. And each year we run a series of virtual seminars like the one we have today. And every 2 years we host a global summit on environmental remediation on the P&L campus, with the most recent summit being held back in November 25. So we anticipate in November 27 having the next summit, which we hope you all can join. As Carolyn mentioned, all the materials and presentation materials and recordings from all the REMPLEX events are available on our website if you're interested. And again, as Carolyn mentioned, that includes today's briefings. So Kelly, if you want to post that link to the chat. And with that, I will hand it over to moderators for today, Katie Mueller from P&L and Evan Starr from SURF. Katie?
Thank you, David. I'm going to take over here first.
Sure.
I would like to thank REMPLEX for hosting and co-moderating today's seminar. It's another exciting opportunity for collaboration on an ever-evolving topic of interest. I am Evan Starr, a SURF Board of Trustee at-large member. SURF is a nonprofit organization comprised of industry professionals from around the world who are dedicated to advancing the science and application of sustainable remediation through collaboration, education, and innovation. Our members develop, lead, and participate in SURF's technical initiatives to improve the knowledge of sustainable remediation. Please visit our website at sustainableremediation.org for more information on upcoming events, to sign up for our monthly SURF break newsletters and future webinars, or visit our LinkedIn page for up-to-date activities and networking opportunities. We also have our own YouTube page, which includes a variety of videos from past webinars, training tools, and our most recent video is a leadership panel discussion celebrating 20 years of SURF that was recorded at this year's AEHS West Coast Conference in March. A couple of highlights on upcoming activities for SURF include a recently launched SURF Connections program for SURF members can join today, which is a new initiative modeled on a mentorship program and is a one-to-one peer networking and knowledge exchange. The intention is to strengthen relationships, share industry experience, and build deeper community across our organization. There is a kickoff meeting on June 24th. Please reach out to our president, Paige Molzan, if interested. Additionally, SURF will be participating at the East Coast AEHS conference held during October 19th to 22nd this year at UMass Amherst. Registration is underway. And our ongoing technical initiative is focusing on launching the Restore tool, which is the remedial evaluation and screening tool for optimization and resource efficiency. This is a new SURF-owned tool for conducting simplified site impact assessments at remediation sites, providing remediation practitioners with a way to quantify the full impacts of electricity usage at sites using current and widely accepted quantitative methods and a user-friendly interface. Current subgroups include existing tool reviews of CIFA and SiteWise, updated methodology of development and testing, and platform development. And again, please contact Paige Molzan to join or with questions. And lastly, I'd like to thank our SURF sponsors who help make all this happen. So turning back to today, I'll let Katie introduce our seminar focus and our first speaker. Thank you.
Thanks, Evan. Today's seminar topic is on enhanced natural attenuation, pathways to sustainable remediation. Our three speakers will explore enhanced natural attenuation technologies and approaches, their associated resource demands and impacts, and methodologies for evaluating and comparing the environmental footprint of different remedies. Our first speaker today is Brian Looney from Savannah River National Lab. Brian is a research environmental engineer at SRNL and an adjunct professor at Clemson University in South Carolina. For more than 40 years, he has coordinated applied research, development, and deployment for environmental characterization and cleanup methods. His work encompasses subsurface access techniques, remediation methods, characterization tools, including horizontal drilling, bioremediation, and tracer testing. Currently, Dr. Looney supports technical activities related to attenuation-based remedies and serves as a senior technical advisor supporting the DOE Environmental Management Program. Brian will first present 3 perspectives on enhanced attenuation. Go ahead, Brian. Thank you.
Great. Thanks, Katie. And thanks everyone for tuning in. This is an important topic. It's one I have a lot of passion and dedication for. So I have a great appreciation for both SURF and Remplex in putting this on. What I'm going to do is try to set the stage and talk about enhanced attenuation. And I'm going to provide, in order to organize a short talk, 3 quick perspectives that lead from kind of the initiation of the underpinning science to a case study. And in order to be efficient about doing this, I'm going to build the perspectives around 3 specific documents. The first document is focusing on the science and the opportunities associated with enhanced attenuation. In other words, what is the technical basis for a sustainable enhanced attenuation or enhanced natural attenuation? The second perspective is, once you have those ideas, you need a way to implement them in the real world and to meet expectations of both regulators and stakeholders. So the second document is a document from the Interstate Technology and Regulatory Council. It's a technical and regulatory guidance document on how to implement enhanced attenuation. And then the third perspective is a case study, which is an exemplar of several case studies that we have done, in our group. But it's just an example of how you actually step through the process and the documentation of performance and sustainability. So in terms of the technical underpinnings, this first perspective, one of the things that has been really helpful to us in interacting with the regulators and public is to come up with some clarity in terms of the anatomy or the structure of contaminant plumes in contaminated areas. And this diagram, this kind of fried egg diagram, is one that has been very effective. And what you see is you have the facility, you have a near field disturbed zone where you have highly perturbed conditions and high levels of contaminants. Then you have an impact zone. This would typically be something like a groundwater plume or kind of contaminated vadosone area. And then you have this blue zone or the transition and baseline zone. Now, the value of this is you can define the characteristics which assign the portions of the plume to each of these zones. Then you can develop the characterization and remediation strategies or the behaviors you have for both the red zone, the green zone, and the blue zone. Now you have something with great clarity that you can talk about with your stakeholders and regulators. So this is kind of the structure. In fact, at Savannah River plant or Savannah River site where I'm located, we actually, with our regulators, developed one of these diagrams for every single one of our 58 waste sites. And then that was the structure of how we focused our discussions and came to agreements on what remediation, and it really helped move things forward. But this is just a snapshot, and what you can see in the real world with time is you start with a source, and then the contamination exits that source. You have a source mass flux that gets into the surrounding environment, and you slowly form this plume. If there are attenuation processes, then there are things that ultimately will stabilize and shrink the plume. Now, I'm going to zoom in. So when we make these diagrams, what we have is a series of remediations or mitigation actions that have several different classes. So this is our continuum, and it goes from kind of the source remediation and treatment on the left to monitor natural attenuation on the right. And you can kind of pencil in all the different technologies underneath. So you basically have kind of aggressive technologies on the left, like excavation or in situ chemical destruction or thermally enhanced removal. So those kinds of things for source zones. Then you get into this kind of interdiction and active remediation, pump and treat, recirculation wells, in situ bioremediation. And then you have permeable reactive barriers, phytoremediation, and biotreatment systems, all the way over to monitored natural attenuation. Now, enhanced attenuation is a really interesting opportunity because what it is it's something that can be applied really across the entire middle part of the spectrum, and it goes into how you design or think about the implementation. So just going back to this, what we're going to focus on for now is the fact that plumes kind of expand, and then if there's attenuation, they shrink, and we need to understand the science associated with that. And then this is a diagram that's from early EPA documentation on natural attenuation, and they showed, again, this is their version of, you have a source, you have a kind of a period of expansion, a period of stability, a period of shrinking, and then ultimately the plume will become exhausted. Now, it'll make a good question for the end of the seminar, but the real world is a little bit more interesting than that. This is a bit oversimplified, but I think it does capture the essence of what we're talking about is plume mass balance or plume stability. And the state of the plume, whether it's expanding, stable, or shrinking, is entirely dependent on the mass balance. When the attenuation within the plume footprint is equal to the source mass flux, that is a period where you, in essence, have a stable plume. If the source mass flux exceeds, then you have an expanding plume, and if the source mass flux is less than the attenuation, then you have a shrinking plume. And that's depicted in these simplified block diagrams where the block is kind of a plume footprint, and you have two mass balances. You have a water mass balance, and the size of the arrows is intended to kind of just be a gross representation of quantities. So into this block, from a water perspective, you have inflow, you have some infiltration, and then you have outflow. And you can see the out arrow is slightly larger than the in arrow because of the size of these. So that's what happens with water mass balance. With contaminant, it's a much more interesting thing. So you have contaminant loading, so you have a source mass flux, goes into this plume, and then you have some flux that comes out to a receptor location. If you have minimal attenuation, then basically the size of the arrows coming in and out are roughly equal. If you have moderate attenuation, you have the size of the arrow coming out being smaller. And if you have enhanced attenuation, you can try to drive the size of the output arrow to an even smaller amount. And the basis of the need for enhanced attenuation has to do with your remedial objectives and what you're trying to achieve. So if we take this apart further, this is structured kind of like a Sankey diagram. And again, the width of the arrows is proportional, in this case, to mass flux. So you have the source zone, and you have this big, fat, wide red arrow, which is your source mass flux going into the plume. And then each of these side arrows represents a class of natural attenuation processes. So you can have things like chemical attenuation processes. So this would be kind of sorption and abiotic degradation, the reaction of the contaminants with, for example, mineral phases and things like that. You have physical attenuation, dispersion, diffusion, and advection, which are weak attenuation mechanisms and not entirely kind of satisfactory, but need to be thought about in context with the others. You have a big one for organics, or your biological processes, both anaerobic and aerobic processes. And then ultimately what that leads to is a flux to some compliance boundary or a calculated risk associated with that flux. And as you can see on this diagram, our arrow is wider than the kind of acceptable amount. So even though there's natural attenuation going on, it would not meet remedial objectives for this site. And so what is enhanced attenuation? Enhanced attenuation is you try to take steps to adjust the attenuation rates in a sustainable manner so that you achieve your goal on the right side of this diagram. And so what you can do is you can do some sustainable actions that reduce the source mass flux so that arrow coming into the plume becomes smaller. You can enhance some of the chemical or biological attenuation processes. And so what's the beauty of thinking about the condition in this manner? The beauty is that each of these arrows to the side becomes a control knob. And I think having the idea that you have multiple knobs that you can control and that you can control them in a variety of ways is really what leads to innovation and the potential for great value in thinking about sustainable enhanced attenuation processes for these sites and being able to transition in many cases from active to passive remediation. So that's really the basis. The basis of the whole idea of enhanced attenuation is really a mass balance and having the availability and the innovation to adjust these control knobs and control variables. Well, that's where we were a number of years ago, and we began interacting with the Interstate Technology and Regulatory Council, and they formed a team to specifically look at enhanced attenuation and to keep things in control. The initial focus was on chlorinated organics, but the ideas, the mass balance ideas and the ideas in this guidance are totally portable and can be applied to a wide range of contaminants. The enhanced attenuation team was state regulators, federal regulators, other federal agencies, specifically EPA and USGS, Department of Energy, universities, and industry scientists. So it was a really nice, broad team. And we developed this document that outlined how to implement and what the definition of enhanced attenuation was. So I'm going to delve into a few definitions. So this definition of monitored natural attenuation is really goes back to John Wilson in the late 1980s. And natural attenuation is, monitored natural attenuation is a remedial strategy that relies on naturally occurring processes in soil and groundwater environments that act without human intervention to reduce mass, toxicity, mobility, volume, flux, or concentration of contaminants. So some of the key words in there are without human intervention. The objective is to use multiple lines of evidence to demonstrate that an identifiable attenuation process is occurring at rates that support achieving remedial objectives in an acceptable time frame. Well, this was great, and it actually was highly successful, but there was a problem at a number of sites. And what happened was the regulators kind of wanted us to have several years of data to, for example, document plume stability. But at a lot of sites, we had already begun a pump and treat. And so the first thing, people would run out and then they would put in a pump and treat. And then the plume was under the influence of the pump and treat. And so you couldn't actually gather the data that EPA in the way that EPA suggested to get information on plume stability. And it was kind of a classic no-win situation where, you know, we want to go to natural attenuation, but you can't go to natural attenuation because you can't get the data that you need. And for those of you who are Star Trek fans, this is a no-win situation, or what they called the Kobayashi Maru, right? So Captain Kirk had to decide, you know, whether to, save the universe or destroy his ship and his crew, right? Well, what did Captain Kirk do? Well, he actually reprogrammed the computer. And so he was the only one who ever beat the Kobayashi Maru. And so enhanced attenuation is really a way to reprogram the computer. And the definition of enhanced attenuation is an active intervention in a subsurface system to increase the magnitude of attenuation processes. In other words, adjust these control knobs. It relies on enhancement that sustainably, and I've underlined that because it's really important, manipulates attenuation processes, leading to a lowering in mass flux of contaminants to meet remedial objectives. And the way that the ITRC defined it is a bridge between source zone and plume treatments and M&A, or alternatively, if M&A was not being effective, it was an opportunity to have a bridge back to some kind of an active remediation. And so the cover of the document is kind of source and plume treatments on the left, monitored natural attenuation on the right, and enhanced attenuation is really a construct that allows you to in a disciplined way, move back and forth as needed between those two areas. This is an early form of, I think, what you could call, adaptive site management. The core of the ITRC was a flow chart and a little miniature, which we're not going to read on the left, But what I've done is I've kind of broken it into zones and I've blown it up. And so what you can see is at the top, you have your normal response actions to an environmental contamination scenario. You collect data on the contaminants, the risks, and the plume stability. And that goes into this group of circles on the left. Then there's a whole series of questions to determine if the requirements of monitored natural attenuation are met. And if the answer to any of those is no, you go into a new section, which was not in the previous regulatory kind of paradigm. And that is there an enhancement possible? And if the answer is no, you basically are in just a standard active treatment and you just circle back up. And if the answer is yes, you determine how to design, you implement, and then you, in essence, go in and then episodically you go back through and you start asking this series of questions to see if M&A is met until you ultimately achieve that. I've blown up just a portion of this area that has to do with the questions on M&A, and you can see are there the normal ones? Are the risks acceptable? Is the plume stable or shrinking? So if the answer is no, that the risks aren't acceptable, then you would go over here, right? You actually ask all these questions. Is the plume stable or shrinking? If it's not stable, you say no. If it is, you say yes. Are the conditions sustainable? Is the time frame okay, et cetera, as you go down. On the right side, this is really the interesting new meat of the flow chart. You basically state the goals of the enhancement. In other words, the goals of any enhancement have to do with what sent you there. So for example, if the time frame was too long, the enhancement would focus on acceleration. If the risk was too high, the time frame would focus on protection. If the plume is not stable, the time frame would focus on stabilizing the plume, right? Then you look at your mass balance arrows and you determine whether there are sustainable enhancements that are viable. If the answer is no, again, you circle back up to a standard active remediation. And if the answer is yes, they are possible, you implement and then monitor those enhancements, and then you cycle back up, but you're now monitoring it as an enhanced attenuation rather than a straight active treatment like a pump and treat or an active bioremediation. Now, what does that lead to? It leads to great potential diversity and variety and innovation in what can happen. So for example, you can intercept water and you can reduce the amount of water flowing through a site. You can collect water at the surface. You can intercept groundwater. You can do groundwater bypass. You can, in source zones, you can reduce the mass flux to the groundwater. You can remove material. You can reduce the amount of water flowing via ET. You can put a downgradient permeable reactive barrier. Again, sustainability is the key. So whatever you do, you now do with the lens and the calculation and the engineering associated with sustainability for the required time frame. When you get into the green zone, this is the red zone, the green zone, you basically again have the potential for trees, for controlling evapotranspiration, nutrients for bioremediation, enhanced bioremediation processes, and a whole wide range of things. When you get to the distal portion of the plume, again, you can have plant-based methods, biological methods, abiotic methods, permeable reactive barriers. But now with your root zones being kind of very near the groundwater, you can blend in some of the rhizosphere processes and the bacteria that happen there. We're going to hear more about some of these things in the other talks today, and I'm really excited to hear that. But what you'll see is just two examples in the other talks of the diversity and breadth of ideas that can be cranked into an enhanced attenuation. So let me get quickly into a case study. This is a former landfill at the Mound site in Ohio from the Department of Energy. It's a 2-acre plume contaminated primarily with TCE and PCE other solvents. The regulatory objectives of the enhanced attenuation were to accelerate the progress, to assure the plume was stable or shrinking, and to demonstrate that the EA remedy was sustainable and durable. What we did was we developed approach using a combined anaerobic aerobic structured geochemical zones. And I'll go into that in a minute. But this is just a kind of a timeline, and what you can see was about 30 years of disposal. Then they did an encapsulation process. They made what they called the burrito, which the bulk waste was wrapped up in and kind of just stored on site. So it was unsightly, and so the public wanted that taken away. So basically in the late 2000 aughts, that burrito was actually moved off site. But during the interim, there was an soil vapor extraction and then a groundwater pump and treat, which was ongoing. And in the about 2014 to 2016 timeframe, we developed the idea to convert this pump and treat to an enhanced attenuation. And the first step of that was a treatability study. Key feature here was the pump and treat was projected to operate until 2040, and the enhanced attenuation actually accelerated the achieving of environmental goals to 2028. Structured geochemical zones is a very simple concept. What you have is aerobic degradation is faster for chlorinated solvents with fewer chlorines on them, and anaerobic is faster for things with more chlorines on them. And so what you have is at a lot of sites where you try to do anaerobic, things slow down and you get accumulation of some of these daughter products. And so the idea was to make alternating zones of anaerobic and aerobic so that anything that was formed in the anaerobic zone would degrade in the aerobic zone. If there was any residual, you'd go in another anaerobic zone and then another aerobic zone. So very simple idea. And what happens in the source zone, we also put emulsified vegetable oil, which spread out on the water table surface, and that provided additional partitioning barrier, so it reduced the source mass flux further and preconditioned the groundwater for the near field anaerobic zone. In that zone, you have some degradation of parent solvents. Then you have things coming off like dichloroethylene, vinyl chloride. Those degrade rapidly in the aerobic conditions. You also have fermentation, so you have methane, propane, ethane, and other things coming off that zone, which feed into co-metabolism in the aerobic zone. And so what you want in the enhanced attenuation is to track all of these things. Here's just a quick diagram. We put the neat oil in under the former source area, and that just spreads out under that former source area. And then we inject emulsified oil in multiple zones along the pathway down gradient. And then this is just photos. This photo on the left is actually T area at Savannah River, but we did the same concept at T area. It was also successful there. But you can see it's just simply vegetable oil totes that we just gravity fed into the deep Veda zone and then spread out on the water table. And then the sequential anaerobic zones below the water table along a long sect of the plume, down the center line of the plume, we used totes of emulsified oil reagent. And here's just a map, and you can see the green circles are the areas where we put the neat oil. And then the blue circles, you can kind of see this area of the blue circles here. So that's our anaerobic zone, kind of an intermediate zone that's more aerobic, and then another anaerobic zone, and then another aerobic zone. So you can see the structure of what the plan was over here. There's many references for all of these things. Again, I'm building it around these three central references, but we collected data in the circular treatability study for 5 years, and we collected data on contaminant concentrations and trends. We collected data on geochemical conditions. Again, you can see kind of the anaerobic zone, aerobic zone, anaerobic zone, aerobic zone, anaerobic zone. And then we collected a lot of data, and for this kind of thing, we went to Microbial Insights and had the array of solvent degrading both anaerobic and aerobic organisms kind of profiled over time. And so you can see for our individual wells, we had really lots going on. And I don't have time to go into all the data, but basically what happened is we had about a 1 to 2.5 order magnitude increase in the organisms that were important to us. And that was sustained and has been sustained not only during the 5-year treatability study, but there's been another 6 years since. We've continued to collect data, And we, in essence, have a stable system all the way through present day. And at the current time, all of the contaminants of concern in all wells are below MCLs with only 1 or 2 exceedances each sampling period. And we projected that all wells for all contaminants would be met in 2028. So we're basically right on schedule for meeting that. So we completed the circular treatability study. We implemented it using the ITRC, and we developed technical guidance. We monitored using multiple lines of evidence with contaminant and plume trends, geochemistry, microbial community markers, consistent with EPA guidance. It's performing exactly as we projected. Last year, there was a formal ROD amendment to officially discontinue active treatment and pump and treat and move to passive attenuation-based remedies. They formally made it into an EA. There's only monitoring and stewardship plan to moving forward. There was a significant cost savings. The most important thing was the acceleration, but there was a $6 million cost savings for this one site. This was the second successful implementation of the structured geochemical zone strategy with the T area at SRS. Now, just one quick final comment. It was interesting, the title of this seminar is Enhanced Natural Attenuation. When we were first thinking about this in the 1990s, we actually developed our initial writings, and we called them Enhanced Natural Attenuation. And I sent the information to John Wilson at EPA, and I said, we have this idea and we want to kind of build this out. And John Wilson said, you can't do that. And I said, well, why not? And he goes, well, natural attenuation is natural attenuation, and enhanced is enhanced, and they can't be the same, right? And so he goes, you can't do that because it has human intervention. And I'm like, oh, okay. So I thought about it overnight, and I called him back and I said, well, we'll just take natural out and we'll call it enhanced attenuation. He goes, oh, yeah, well, you can do that. So we've been pretty careful to call it enhanced attenuation. It does not hurt my feelings to call it enhanced natural attenuation, but I just wanted to share that anecdote that that was John Wilson's response when we first kind of pitched it to him. So the 3 perspectives that we've talked about today are understanding and characterizing using a mass balance approach. That's kind of the underpinnings and leads to creativity and innovation. Doing things in a disciplined and organized way, similar to what ITRC has put in their technical and regulatory guidance. It has metrics and contingencies and has multiple lines of evidence as kind of a requirement of monitoring. And that EA has the ability to provide a bridge to transition to attenuation-based remedies and has been successful in multiple case studies. And then these are the links for the 3 reports I put in there, and they will be on the Remplex website. So that's all I have, Katie. Sorry if I went over.
No, thank you very much, Brian. We will be taking questions and answers at the end of all the presentations and much more open discussion. So please, if you have any questions, feel free to enter them into the chat and we will get to them after the presenters. So next, let me introduce our next speaker. Maria de Lourdes Diniz is a professor in the Department of Mining Engineering at the University of Porto, Portugal. She holds a PhD and an MS in environmental engineering from the University of Porto, as well as a 5-year degree in mining engineering. With 25 years of experience in environmental issues related to radioactive contamination, her work focuses on the environmental impacts of uranium mining, environmental restoration, remediation, and sustainable approaches for the treatment of contaminated water. Currently, her research explores bio-based adsorbent materials for the removal of uranium from aqueous solutions, with an emphasis on environmentally responsible and cost-effective strategies for real-world applications. She has also collaborated with NATO and the IAEA for several years on capacity building projects. Today, Maria will present on sustainable remediation of uranium mining sites. Take it away. Thank you.
Hi. So thank you, Evan, for the introduction. So today I'm going to talk about environmental remediation technologies applied to water treatment by using these green adsorbents. So just a little of background. Environmental remediation technologies plays a crucial role in addressing the environmental liabilities, ensuring that contaminated sites are effectively treated and maintained. And the efficient and long-term performance influence the need for ongoing surveillance and maintenance. And many times, the adopted solution the solution that was chosen at a certain point or certain time is not working anymore or doesn't work at the long term. So it's very important to have this result about the efficiency and long-term performance. Usually, the environmental remediations are characterized into conventional and emergent methods. So many of the conventional methods have demonstrated to be effective in specific scenarios. And these approaches are often employed or widely employed because of their established protocols and predictable outcomes. So it's already known that they are effective. Nevertheless, they perform or they have significant limitations in terms of scalability, cost, performance, and also long-term sustainability. For the emerging technologies like bioremediation, phytoremediation, and nanotechnologies, these offer promising alternatives able to degrade or demobilize contaminants. Yet their performance can be inconsistent because of their site-specific variables and also limited field validation. On the other hand, advanced remediation techniques have expanded the range of available technologies, and these methods enable more targeted and efficient interventions in particular in complex contamination scenarios. Nevertheless, despite their advantage, there are still several challenges to be addressed related to high energy consumption, the risk of generation of secondary pollution, and also stakeholders' acceptance persist. Many times the public perception and the regulatory hurdles, they often impede the deployment of novel technology, especially when risks are poorly communicated or the benefits are not clearly demonstrated to the communities. So despite of these huge advancements in remediation technologies, there is still a growing need for an integrated multi-technology approaches that combine physical, chemical, and biological methods to enhance the overall effectiveness. And this is where our work fits, as we are studying a train of water treatment steps with different technologies to apply to mine water or mineral beneficiation wastewater. By now, we are considering 3 or 4 different treatments to apply to this type of water, and we are focusing in particular in the use of natural and waste-based adsorbents. Adsorption is a widely used traditional technology to remove radionuclides from water because it's a simple process, it's simple to operate and cost effectiveness. The high efficiency of the process is attributed or due to the high surface integrity of the adsorbents of these materials, and the chemical and mechanical stability, and also the distinct pore structures, because they are very particular structures that can trap the contaminants. These materials can be derived from recycled sources, including forestry, agriculture, and biological sectors. And on the other hand, these green adsorbents align with the circular economy and offer a sustainable alternative to water treatment in contaminated mining sites. So these green adsorbents have emerged as a potential alternative to conventional synthetic materials used in water treatment process. And this interest growing, or this growing interest in the green adsorbents stems from the need to address several limitations of synthetic materials. Nevertheless, there is still the need for large-scale applications, which requires that the raw material be abundant and inexpensive, so low cost, and that the process be fast in the adsorption itself. And in this context, there are several types of green adsorbents. They commonly include bio-based carbon materials such as biochar and activated carbon. Activated carbon is quite or widely used. These are derived from agricultural residues. There is also fibrous plant materials such as coconut ash and sawdust. Also green synthesized nanoparticles using plant extracts or microbes. They offer high specificity in contaminant removal. And natural clays and minerals like bentonite and zeolites, they are also widely used because they provide high surface area and also ion exchange capabilities to pollutant removal. So our target with this work was and is still ongoing to develop the green adsorbents to be applicable to mineral processing wastewater, so active processing plants, and also water contaminated with NORM, acid mine drainage, and mine water treatment at remediated sites. And this is a case study that we are using here in Portugal. That site that is already from a uranium mining site that was already rehabilitated, so remediated, but there are still ongoing water treatment activities. So we target these for different materials. First, to remove radionuclides, chitosan, modify bentonite, also modify the attenuation earth and moringa seeds. Then we want to test for the other contaminants in the water, so heavy metals and organic pollutants, so for the regulated and non-regulated contaminants. From the literature, there are a few studies with these materials, and it's quite interesting the results that are described in the bibliography. For the moringa seeds, some studies report an efficiency in removing uranium above 95%, but also for metals like manganese, nickel, arsenic, cadmium, and lead, with the modified to-measure first, an efficiency higher than 90% for uranium thorium. For the modified chitosan, so this is produced with the shellfish waste, very cheap material actually, so it's a byproduct can be a byproduct from waste. It showed an efficiency above 90% for uranium, thorium, and lead, and the modified bentonite also with very high efficiency for uranium and thorium with 90 and 95%. So our exploratory work target these materials with that were developed in batch experiments with modified diatomaceous earths and also with bentonite, modified bentonite, with crushed moringa seeds and the raw chitosan. So for the results that we achieved with the modified diatomaceous earth, we used 3 different uranium concentrations in artificial waters for these exploratory works. So we tried to test at low, medium, and high concentrations. And for the modified diatomaceous earth, the efficiency was nearly 100% at the lower concentration and 93% at the higher concentration of uranium. For the crushed moringa seeds, the efficiency was 100% for low concentration, 84 for the medium concentration, and 80% for the higher concentration. So there's a need to optimize the parameters that are involved in the experiments. For the raw ketosane, we have a very few efficiency compared to the modified diatomaceous earth and the moringa seeds, about 30%. And I will explain later what may have caused this low effect. But for all of them, the contact time is extremely low. So between 2 and 4 hours, it's possible to achieve this very high, very good efficiency, removal efficiency. For the modified bentonite, the results are not so good because the bentonite formed, it has a swelling effect. It was very difficult to filter this material, so we abandoned the bentonite, so not working with this material anymore, and focus on the moringa seeds and the modified diatomaceous earth. So from the literature on moringas, on application of moringa seeds as adsorbent in uranium removal, there are reports about 97% removal of uranium from synthetic solutions, 87% of uranium removal from acid mine drainage, also effective with metals, between 90 and 100% test with synthetic wastewater. It has also been used as natural flocculant in water purification, with efficiency about 99% for colloidal matter and between 90 and 99% of bacterial removal. In complex wastewater comparable to industrial effluents, very high efficiency, also between 92 and 95%. And in water contaminated with pharmaceuticals, also efficiency above 95%, but at least we couldn't find any studies of application of moringa seeds to mine water or mineral processing wastewater. So what concerns, which is good because for us is a novel application. So it's a new work that is being developed with this material as a green adsorbent. For ketosane, there are studies in the literature that report an increase on the efficiency of this material to absorb uranium, but rising from 29% at pH 2 until 90% with the pH 5. So the pH is a key player in the adsorption efficiency for ketosane. And in our case, we test a pH 2, so that's why it didn't work. The efficiency was very low. For the attenuations, modified attenuations earth, so the studies that are reported in the literature show a very good efficiency in removed uranium, starting 72% at pH 3, and then it increased until it could increase until 96% with a pH 8. It also has a very good effect, as I mentioned before, to remove thorium. This study at pH 3, it was possible to have 90% removal, but it also with cesium, chromium 3 and 6, lead, but this is highly dependent on the pH value. So we proceeded with our work, trying to optimize the operation conditions. We developed a fractional factory plan for the 3 levels, low, medium, and high, applied to 5 variables. So pH, temperature, adsorbent concentration or adsorbent dosage, initial uranium concentration, and the contact time. And we were already able to optimize these conditions for the attenuation earth. And so the adsorption model is identified, and now we are ready to scale up this work. For the moringa seeds, the optimization work is still ongoing. So for as a summary of these results, we have a low efficiency for unmodified ketosane because we use pH 2. Very good extraction with moringa seeds up to 99%, but this, as I mentioned, is not under optimized conditions. But for the detumations, modified detumations first, the optimized conditions allow us to achieve 87% efficiency. So we are able to validate these materials as green adsorbent with our results and based in what we already described in the literature. And these green adsorbents are capable of addressing radionuclides and also complex heavy metal contamination. So different matrix of contaminants. Some of these results are under publication and being applied to the beneficiation being applied to the wastewater of the beneficiation of rare earth elements from our studies. So to conclude, we may say that green adsorbents that we are studying achieve very high removal efficiencies for uranium and thorium. We only test for uranium, but also for thorium based on what is described in the literature. These materials maintain efficiency after multiple regeneration cycles, so they indicate strong economic potential because they can be reused several times. There's also a higher public acceptance because it's perceived environmental benefits, lower long-term costs, and alignment with sustainable practices like circular economy and zero waste. And we need, but for the laboratory success to be validated, we need to move forward to the field tests to evaluate the scalability and the long-term performance. So the next step for our study is to move for a pilot scale and field validation across diverse contamination scenarios and sites. So our materials should be applicable to other contaminated sites in order to have a diverse application, not only a batch test or lab test. So we really want to move forward with this study. And I think this is the last slide. So thank you so much.
Thanks, Maria. Please continue to put your questions in the chat for the Q&A session at the end. And now we'll turn to our final speaker, Chris Gale. So Chris Gale is president of Applied Natural Sciences, a North Carolina company that specializes in phytoremediation. Mr. Gale has worked on dozens of phytoremediation systems across the United States and in the European Union. He has over 20 years of experience managing and implementing a wide variety of environmental site investigation, characterization, and remediation projects, including all aspects of building and leading successful teams on large-scale, complex problems. Mr. Gale also has experience with a variety of remedial technologies, including in-situ bioremediation, in-situ chemical oxidation, reduction, thermal treatment, soil vapor extraction, and dual phase extraction. So Chris, I'll turn it over to you.
Thank you very much. So I'm going to talk about, I appreciate that our previous two speakers have already on the really focus more on some of the plant-driven enhancements of some natural attenuation processes. Go through basic reminders or introduction to the mechanisms by which phytoremediation work. There's fairly diverse. Also some phyto-enhanced natural attenuation that we've seen and how the plant-based systems can function. Introduce you to our tree well systems because some of the case studies that I'm going to show are fairly unique to applied natural sciences and our tree well systems, and it's a little bit of a different application of a phytoremediation system. And then some of the data that we demonstrate how these systems are functioning. So the phytoremediation mechanisms, there's a few different mechanisms by which phyto can work. So phytotechnology, so phytosequence really just it's within the root zone or to the plant, to the roots themselves, or within the so rhizodegradation, a bit on that little bit later. So that's, I think Brian touched on that a bit earlier. So some microbial or fungal degradation. So it's a lot of times what I see is hydraulic. So that's uptake of water by plants. So the plants are being used to provide hydraulic control. Phytoextraction. So that's actually slightly different from the phytosequestration. So where the contaminant is actually being removed from the subsurface from either shallow soil or groundwater and pulled up into the plant matter itself. Well, also a bit later on phytodegradation. So we actually degradation occurring within the plant tissue itself. So we'll go through the mechanisms by which that can work. So phytodegradation, that's also another one that I see oftentimes is for a lot of organics that are taken up and removed from the subsurface, can be translocated through the plant and then the volatilization transpired into the atmosphere, where then a lot of these organic compounds can be degraded by UV. So I'm going to talk quickly about phytodegradation, the process here. So in-plant degradation occurs by a variety of different mechanisms. So there are some oxidizing and reducing compounds that are produced during photosynthesis. So you can get some natural oxidizing and reducing reactions happening within the plant themselves. And also the endophytes, so some bacteria that live within the tree tissue or the plant tissue itself that can actually physically degrade certain compounds. So as those compounds are taken up from either the groundwater, shallow groundwater, or the soil water, so removing it from the soil or groundwater, and pulled up into the tree or a plant, where then they're often degraded. Rhizo degradation, so that occurs through the tree or plant is providing some root exudates that help to stimulate the local microbiome. So this is from ITRC document run phytotechnologies from a few years ago. So really the plant is providing extra sugars or amino acids, organic acids, some metabolites, other signaling molecules, sometimes some inorganic ions like hydrogen to help balance pH. So mucilage, some polysaccharide-rich coatings, proteins and enzymes and tannins. Many of these compounds help to provide nutrients or energy to soil microbes or fungi that can actually then do the heavy lifting of degrading the contaminants right around the root zone. So typically this is right around the root zone, a few millimeters. The root exudates don't travel much further than that. But what I've seen with our systems is we have some much more extensive degradation that occurs. And so I want to introduce the tree well system to you. So what we do is we drill with a large diameter auger, typically 42-inch to 60-inch auger, down to the impacted groundwater zone. And this is really focusing on a groundwater technology. And so then we put up the green here is what we call a root sleeve liner. So it's a plastic liner. So we'll drill down to the target zone, put the liner in the hole, and then backfill that hole with a loamy sand amended with organic matter, typically 5 to 10%, because we want to provide nutrients for the tree that we're planting in the tree well unit. And then once the tree's planted, we close that liner up around the base of the tree. So really, we're encouraging and forcing downward root growth. And so really the only place the tree can get water is from that impacted target zone. And what we see is that as we draw a lot of water upwards through that soil column, because it's amended with organic matter, we see a lot of degradation occurring for a lot of organics, so chlorinated solvents, petroleum hydrocarbons, 1,4-dioxane, for instance. And so what we're seeing here is that the tree basically is acting like a solar powered pump, pumping groundwater into an in-situ bioreactor. We actually have evidence from a number of sites that we'll jump into here shortly that show significant contaminant degradation, even getting into the rhizosphere or the roots, or degradation within the tree itself. So I'm going to go through a couple of case studies here. One that we look at for a 1,4-dioxane site. The next one we have was a mixed DNEPL chlorinated. It was And then we'll kind of jump into the microbial analysis. So we see some of this degradation, and then we wanted to look into the processes and what microbes may be present that are actually doing this work for us. So this first site is actually produced 1,4-dioxane, had concentrations in groundwater of milligram, very, very high concentrations. The initial objective of this was to really provide hydraulic containment, so capture the down gradient plume prior to it discharging to a surface water. And the idea was the initial thought was that it would take up the 1,4-dioxane, volatilize it, and then it would ultimately UV degrade within a few hours of sunlight. And after the second growing season, we see that, okay, we are proposed that hydraulic containment. You can see this is during the active growing season where the plume was actually pulled towards the trees. The trees couldn't be planted along the plume because there was an active over the plume. You can see pulling the plume towards the trees. So we wanted to dig into this and say, okay, are we seeing this volatilization? So I actually went and tried to measure 1,4-dioxane concentrations in off-gassing coming from the trees. And so a few years, a number of the trees, and determined that less than 1% of the mass that was estimated to be entering into the phytoremediation system through groundwater was being transpired to the atmosphere. Subsequently, looked at tree tissue sampling, tried to find 1,4-dioxane within the trees, and again, see very very minimal concentration. And then looked into the rhizosphere and into the soil column and see substantial evidence of in-situ degradation occurring. This is some work done by others, not done by me, and I don't have a lot of the data to share, unfortunately. But they did identify a fairly robust population, a consortia of bacteria that were degrading the 1,4-dioxane in situ. And even with the 1,500 milligrams per liter in groundwater, there were no phytotoxic effects seen at any of the trees that were planted here. So the next site, this is our site. So it was a waste pond that was chlorinated, including DNAPL. So we looked at a couple of different units. I'm really going to focus on the three that are on the left side here. We have these straw units that help us get down to deeper groundwater. We're able to use phyto at this site down to 115 feet. But what we really want to focus on are the three on the left. So we wanted to do soil, and then we were concerned about potential phytotoxic land above a DNAPL. So we included another set of these trivial units that included addition of ZVI, so zero valent iron, and then a third that included a combination of both ZVI and Daramin, so to try and get some abiotic degradation and reduce those concentrations before it got to the tree. So what we're looking at here consists of total VOCs in groundwater, and this is it is in a log scale in milligrams per liter. So the top kind of purple line with the closed diamonds is water entering the bottom of the system. So entering from the formation, you can see in the hundreds of milligrams per liter. Then going through the ZVI, we get some reduction, and then ultimately the open squares, the red line with the open squares, the concentration that's right at the top groundwater zone, base of the root zone. We see multiple orders of magnitude reduction. And so the question is, because of, is it abiotic or is there some sort of biotic compare that to just the organic matter amended backfill, see a very similar order of magnitude reduction. So these are just two different of these units. The orange closed diamonds and then the orange squares are within the same trivial unit, and then same with the yellow diamonds and yellow squares. So I think the key here is that we see multiple order of magnitude reduction just vertically migrating upwards through that soil column. So there's evidence there that shows that there's some pretty substantial degradation occurring. So because of these sites like these, we see these in multiple sites, chlorinated sites, 1,4-dioxane sites, petroleum hydrocarbon sites, we see a similar pattern. We wanted to look at what is happening, what may be we're trying to identify how is this happening. So we collected some tree tissue samples from two different phytoremediation sites, a chlorinated site and a petroleum hydrocarbon site. Did some next-gen sequencing on the plant tissue DNA to try and identify which endophytes may be present within these trees at these various sites to see what is the process, what processes are at work are helping us within the tree, looking in the tree itself as well. I guess one thing that pointed out to me by the just were doing this work is there were some difficult to understand the differences. There are some complications with seeing the differences between willow mitochondria DNA and some endophyte DNA. So there were some complications there, but I think our team was able to figure this out. I do not expect people to read everything here on the right. This is the results of what we found in the tree tissue. But the kind of take home here was try to summarize on the left is that there are a lot of common endophyte bacteria that were identified in these samples, and a lot of them were active degraders. So we did identify Dihalococoides in the CVOC site, pseudocardia, so 1,4-dioxane degraders. We have aerobic naphthalene and DCE degraders, obligate herbicide degraders. So a lot of these degraders, and we did not inoculate any of these trees. So a lot of these, the bacterial population is kind of self-established. So we were able to see that there's the bacteria are able to grow into these trees by themselves when there's the contaminant mass present. This is another CVOC site. What we're looking at here now is focused on what we see in groundwater. So the blue here is we took groundwater samples from outside of the tree well unit at this site. It is a fairly aerobic aquifer, and trying to look at Dihalococoides and saw very, very little to no non-detect concentrations of Dihalococoides here, whereas within a groundwater sample collected within one of the tree well units here had very, very high concentrations of Dihalococoides. And so I might point out that the piezometer that was in this tree well unit was sampled and I think co-located with another well that was just probably 15 feet away. And so with 2 × 10⁷ cells per liter Dihalococoides within the tree unit and non-detect outside in the formation. And again, these were not inoculated. This is just what was kind of self-established within the tree well system itself. Just so some quick conclusions on this. So I think the plants and the microbiome often work together to degrade many contaminants, a wide variety of contaminants. So endophytes and in-situ microbial populations, we do see that they can self-establish. One of the questions we're looking at is could the addition of microbial populations could be inoculate to further enhance or expedite the effectiveness of these systems. So Bry degradation also is a key mechanisms by which phytotechnologies help to provide remedial function, and is also a key natural attenuation process that is often enhanced by the presence of plants. I think that's all I've got today. I just want to acknowledge my colleagues, Paul Thomas, and some contributors, Jim Linton, Doug Riddle, Jen Wilkinson, and Phil Dennis. So all I have.
Thank you, Chris. We had some minor technical glitches with your video there, but I think it's just temporary. I apologize for the inconvenience there for everyone. But we'll welcome an open discussion, but I'll first start with you, Chris. So an easy one, I think, would be just what are the typical O&M requirements for a phytoremediation system?
Yeah.
Maybe
O&M system, especially the first.
Sorry to interrupt. Maybe go off video just to see if it improves the connection. I don't know.
Your break. I'm having trouble hearing you. Sorry.
So maybe try again with the typical O&M requirements. We're just seeing if going off video might help.
Yeah, hopefully that helps. Is that helping?
Just plow right through it.
Okay, sorry. Not a no O&M system. It's an O&M system. They are, we are dealing with a living tissue, so there are, we have to manage pests and fungal. But typically it's more labor intensive the first few years, but as the system matures and mature, they become more resistant and more resilient. So lower O&M is needed. But typically, kind of a rough order of magnitude, you're looking at a few, less than $20,000 a year O&M to start with, and that's a pretty low operation. But it is a substantial mechanical system.
Okay, thank you. And Brian and Maria, please rejoin us when you can. Katie?
Yeah, so I think this next question will turn to Brian. So Brian, what are some key characteristics that would differentiate how you design an enhanced attenuation remedy versus an active one?
So the key is really the issue of sustainability. So for example, over time, if you actually look at the history of bioremediation, in the early days, the scientists all used small molecular weight laboratory chemicals because they wanted to get reproducible data. So there was a lot of work with lactate and things like that, sugars. Those are they're kind of on the hot end of the spectrum, so they burn up quickly. Over time, what you've seen in the industry is a shift toward longer-lived reagents. And that shift toward longer-lived reagents has had a unexpected, or I don't know, a kind of a beneficial impact in the sense that people started noticing that the impact of the reagent lasted a lot longer than just the quick calculation, and that has to do with sustainability and the recycling of the carbon by the microbial community as the cells die and things like that. So what you have seen is that the robustness and the sustainability of an action are the key differentiators between, for example, an active bioremediation where you can use molasses and sugars and things like that, and they work great. But then you have to go out regularly and kind of reapply them. Then you get into the vegetable oils, and they have like a lot longer lifetime, and then a few legs associated with the cryptic regrowth of the reuse of the carbon. And so sustainability is really the key. And when you design it, what you do is you design, for example, the amount of reagent that you inject to last for the period of time that you think it needs to last. So we designed Mound to add an amount of carbon that would actually be effective through the 2040 timeframe, because that's when the natural attenuation would have actually taken it to where it needed to be. And so those two timeframes correlated with each other. So that's the kind of logic that you use.
Okay, thank you, Brian. And for Maria, I just wanted to ask, what is one of the main ideas or innovation behind the green adsorbents?
So for this type of materials that we are trying to develop and improve, it's because there is technology, the adsorption is already used in modern water treatment, but only a very few adsorbent materials are used at industrial level. It's completely in contrast with the exponential growing of what is described in the literature, because there's a lot of studies at batch or laboratory scale, but at industrial level, there is very few. And in this context, current remediation methods can be expensive and not always sustainable. Again, the sustainability issue is a driving, because having these adsorbents produced from waste or natural materials is sustainable and is reusing something that is not valuable anymore with the case of the waste. So we are trying to explore more environmental friendly and cost-effective approach to clean or remediate contaminated sites. That is the main reason that we are testing these green adsorbents. Instead of relying only on the conventional chemical treatments, this adsorption can be considered passive treatment, as I said because they don't require energy input or operational control to work. So this is the main innovation.
Okay, and then a quick follow-up. There's a couple related questions regarding to how do you remove the green adsorbents from the water, and do they remain? Do they need to be treated, disposed of?
Yeah, so the granule remains bound to the seeds, to the biomass. So we have a granulose spent adsorbent, and this must be regenerated or disposed of as contaminated solid waste. But the material as adsorbent can be reused several times. So, and for each extraction, we need a very, very low amount of adsorbent. So it takes very small quantity of these materials, but at the final, what we will have, it's something that needs to be disposed of course.
Okay, thank you.
All right, we have another question for Chris. So in the chat, there's some discussion around the maximum treatment depth of the tree well system. Can you discuss a few sites where you've evaluated the maximum depth of your system and its ability to treat effectively?
Yeah, that we about 35. That's for unconfined aquifer systems. The systems that I showed with the slide with the straw well system, we've gone as long as hydraulic head in a confined, either a fractured rock or confined, semi-confined system. We've gone to 115 feet effectively, but that has water potentiometric surface around 15 to 20 feet.
Now, for Brian, do you include monitoring during the remediation process to ensure the remediation is working as planned over time? Are there certain parameters that you use for measurement?
Yeah, monitoring is critically important, and we use the multiple lines of evidence approach that EPA talked about in the original monitored natural attenuation guidance. And so we divide the information into categories. So you look at contaminant trends and concentrations and plume metrics, plume stability metrics. We also look at if there's a geochemical aspect to the remediation. We look at the fact that the geochemistry is in the range that would support the active or the ongoing sustainable remediation process. And then finally, we try to collect specific data to document the presence and activity of the attenuation process. So we use a lot of microbial monitoring. And I saw another question, and I can just kind of extend this. So there was another question on natural source zone depletion for LNAPLs. And that's a system that relies on really an subsurface ecosystem and the presence of both anaerobic and aerobic zones, and kind of the depletion of an LNAPL source over time as it's being kind of burned away from the down from the edge, because the aerobic processes are faster than the anaerobic processes. Kind of understanding that dynamic is the whole basis of natural source zone depletion. And so if you were just going to let it happen naturally, you would want to monitor the key aspects of that. If you wanted to enhance it, and I can think of a couple of ways to do it. So first, you might add, an electron acceptor, a very strong electron acceptor. But then you would want to calculate to add an amount that would be sustainable. Another way, kind of a slower but steady way to do it, would be to use. We've developed, and some other people have used, some barometric check valves where you basically can control the flow of air into the subsurface. So if you put a bunch of wells in that let air in around the edge of the plume to accelerate the natural source zone depletion, you would want to add metrics associated with what would be the effectiveness of that, what would be the rates of that, and that it was happening and it was sustainable over the long term. So really just multiple lines of evidence and monitoring the thing that you care about.
Great, thanks, Brian. So We have a few other technical questions for Maria that were in the chat. So do some absorbents work best for some radionucleotides? And what is the removal rate of decay products like radium?
Mhm. So we didn't test or we didn't focus on radio because our wastewater, the water that we studied until now didn't have any radio, only uranium and very few thorium, because thorium is not soluble at all. But anyway, we didn't have radium. But our case study here in Portugal, it will have radium-226, but it may be relevant to consider radium and the other radionuclides because they can be associated with uranium-contaminated water in natural environments, meaning uranium-bearing geological systems. And of course, it will be present the decay chain process. But for mine water, mine wastewater for mineral processing, it may make sense to look into radium-226, but not so to the other radionuclides, because they will not be present. So uranium and radium can be, it makes sense if we are dealing with this kind of industrial contaminated water. For natural contaminated waters, yes, it may be necessary or makes sense to look to the all decay products from the uranium chain.
Thank you. And Chris, are there physical or theoretical limitations on the depth to water for the tree well system?
Yes, there are. So the trees need to be able to root down, and trees, I think I just addressed this a little bit earlier, about 35 feet needs to be max depth for an unconfined system. Confined systems, fractured rock systems, if we can rely on attachment head, we can get much deeper than that. But head needs to be within 35 feet or so.
Okay. And then do you ever inoculate the tree roots with any fungi to stimulate plants or the rhizosphere microbiome community?
Yeah, we have included some mycorrhizal fungi oftentimes when we plant, along with some fertilizer to try and help that. There's been some interesting research recently trying to identify more specifically certain mycorrhizal fungi that may be exuding, for instance, like a vitamin B2 that we could then use in collaboration with some other microbial. So there's some newer research to try and more specifically target that microbiome to certain contaminants.
Great. I think we have time for maybe two more questions. So this one's for Brian. Are there example cases where enhanced attenuation may not be appropriate?
Yeah, I can be very brief on this. So basically in the red zone where the source mass flux is so high, the idea of an attenuation-based remedy is probably not viable because you're not going to be able to get the rates to the place you need to be them. You need more aggressive technologies. So I would say there is a cut point at kind of a very high, so like not for DNAPL in the case of organics, or not for areas where there's actual uranium minerals that are slowly dissolving in the case of like radionuclides, so things like that.
Okay. And Maria, what are the next steps for this research?
So the next steps are, as I mentioned, to move forward, test these materials in more realistic conditions and on a larger scale. We also want to better understand how they perform over time and how they can be reused, these materials. And the goal is to move from laboratory to real-world applications. Nevertheless, the solution development progress needs to have other components like the sourcing capacity, such as kinetics, regeneration, economics factors, or the environmental sustainability. So we need to do all these assessments to have at the end an industrial application responding or fitting to what we call to green adsorbent, low cost, and sustainable material.
Thank you. All right. Carolyn? Katie?
Thanks for all the great questions, and thank you very much to our three fantastic presenters today and to everyone who joined us online. Really appreciate it. It was a great discussion. As we mentioned at the beginning, there's going to be a recording of this seminar that will be posted on the Remplex website soon. And as you leave the Zoom meeting, you're going to be prompted to answer a short survey. Thank you in advance for responding, because we use that information to shape future events. We always want your input into what you'd like to see at these seminars. So please fill that out and stay tuned for information about the rest of our 2026 and 27 seminars. So thank you very much for your time and have a good rest of your day.